So welcome back to Computational Biology Modeling Methods. This is lecture 13. We're going to be talking about explicit forces, persistent motion, introduction to networks. As always, I need to remind you that the class is live streamed and recorded. It's a lot of material to cover today, and so we're going to go through this material rather quickly. There are extra slides in the deck, which will provide additional background material, which will fill in, I think, some additional gaps. Again, we will start out by talking a little bit more about chemotaxis, and then we will move on to doing exercises on persistent movement using the external potential plugin in Computational. And then we will begin to talk about network modeling. Network modeling is a big topic. We'll only get started on it today, and that will be the topic of the remaining lecture portion of this class going forward. Today, we're going to be focusing on the syntax which Computational uses to specify network models, which is a language called Antimony, which was developed by Herbert Sauer at the University of Washington. If we have time at the end of the class, I'll talk a little bit about how to interpret network diagrams, which are the currency of biological models, with our understanding of mathematics to build actionable, executable network models. So I need to remind you that we have the projects due in preliminary form in a week. That's coming up pretty soon. And so I would appreciate people getting ready for that. I know people have been working hard. I wanted to focus today a little bit more on the assignment that you'll have next week, and you may want to revisit it as projects develop, which is that I'm asking for each team to do an in-depth review of one of the other team's projects. And I'm also going to ask people to do short reviews of all of them. So the short reviews you'll write up during the class presentations. The in-depth reviews you'll meet as teams with each other, review the materials, and write up a more detailed critique to help the team you're reviewing improve their models and their representation. Did everybody get the link for the scheduling? I just need a reminder of the team names and composition, and then I will assign pairing up of teams to do the reviews. If you need it, that link is here. I could put that in the chat. No, maybe I can put it in the chat. I'll put it, Juliano, you can probably put the link in the chat for me. That's all right. And I was going to just go over the very quickly the presentation reminder. I emailed this out to people today. This is the review of your classmates' presentations. And so again, when you're doing a presentation, you want to explain what you're doing, why you're doing it, what the key inputs and outputs of your models are, the mathematics of it, and then demo the application, explain the supporting material online, and walk the class through your application, and then show us the code that you use to make it work. And if you've extended the things that you were presented in the source material or changed them in some way, you want to explain that. Explain how you're going to improve your model during the next 2 weeks after your presentation next week. And also, since you put a lot of work into these and you're putting them up publicly, think about how you can publicize your application to increase uptake and interest. When you do your review, you'll meet with your target team, and then you want to be able to first describe what you think the target team's application is, not using their words, but using yours. because you want to understand whether that information is conveyed successfully or not. Ask the question whether the materials that are being shown to you convey those ideas clearly and effectively and accessibly. Our targets are probably undergraduate students or early graduate students, and think about how you might improve that presentation to make it more accessible. Are the scientific and learning objectives of the application clear in the supporting material? I'm hoping that people are seeing the assignment shared, screen shared. Is that correct? Yes.
Yes, we see the assignment.
Okay. If I start talking and I'm not screen sharing, please tell me right away. It's always embarrassing to have to go back and redo things. Not that I haven't been embarrassed in many times in my life over the years. Okay. And then you might describe what the other team is trying to do mathematically, check whether they've cited the sources appropriately, are the figures that they use to support their material appropriate, and are the exercises interesting and useful in terms of the learning objectives? Are there enough of them? Are some of them redundant? If you're doing a replication of a paper, is that replication successful? And then evaluate again the sort of the context of the application. The summary page in NanoHub, is that appealing? Would it inspire you to open that application? Is it correct? Could there be better figures on that home page? Anything that you could do that would be important to increase the usage and uptake of the source material and the applications. And I realize it's a short time frame to do this next week, but we've got to get your reviews back to the people who are doing the projects so that you have enough time to include that material in your revisions. And again, if people want to do a second round of this as the projects develop, I'm sure that the teams will appreciate it. I realize that this is not as standard in science courses, in an art course or in an architecture course, for example, and in many engineering courses, projects are reviewed in this way. People who've taken classes with me before have done this. I don't know if the other classes you've taken at IU have done this, but last semester, people took it quite seriously, and I think the projects got much better as a result of the review. It really made a big difference. And so ultimately, if you want people to help you by reviewing you seriously, it's important that you take it seriously on your side and put the time into it to do a review that's really useful to your other teams. Okay. So that's back to the project schedule and the review assignment. If there are any questions about the review assignment, come back to me or to Juliano. We're eager to make this work. And then again, May 3rd is the very last day. We need final presentations of the revised projects and everything uploaded and running on Nano. Okay, so not everybody's here today, but why don't we just go around the room quickly and get updates on how things are going for people. Logan.
Yeah, Nick and I think the code is pretty much like where it's going to be for like the preliminary submission. Like I was talking about last time, there are just a few like things mathematically, like working out a reciprocal equation so that you can enter in your own IC50 value for a given drug versus just using the one I've put in. So that's the call for the rest of the week. And then the report is something that Nick is working on, that he got the revised code that like with my changes from my labs data today. So he'll go through and do that. And then we've started the NanoHub upload process. And this was the educational exercises and tutorials. I've started all my outlines for them, but I was just confused like how much of a in-depth process you were looking for. Like, are you just looking for me to give like a few examples of like how to modulate the parameters and like what they do? Or are you looking for me to explain like every single part of the simulation and show people like what each part does? Because that like I couldn't really find a good guideline for that.
Right. Well, unfortunately, that's one of these things which is up to you. Let's think about it this way. Ultimately, the key thing here is that you don't want all the work you put into this be lost. And so if you want people to go to NanoHub and use the code, it has to be self-contained enough and interesting enough that people will use it. And so I realize that the more you put into educational materials and support, the more work it is for you. And there certainly are a lot of details. You don't need to document every line of code. But if the key ideas that are presented are not clear, then people won't use the code. And so maybe think about it this way. If you were going to use this application and the supporting materials online as part of a job application, if you were applying to Google or Facebook, how would you want it to look? And then you have to decide how much effort you want to put into that. I bring those up because there are people who have gotten jobs at those places based on these projects. I'm sure you have other things in your life besides that, but when they're good, you could get a fair amount of leverage from these. But you have to decide, given the other demands on your life, how much of your time you want to put into it. And of course, you're also free after the class is over to go and update it and improve it if you feel that that's something that would be useful to you. It's not a static thing. So you have the ability, if you think that that's something that would be valuable to you, to keep doing.
Right.
Again, the goal of all this, I realize doing it this way is more work for people, but my goal is so that the time you put into it isn't wasted. That's really the key thing. And by all means, that's one of the reasons we're doing this review now, because the team that's doing the review can say, hey, you really need more information about this, or you have enough information about that. And that's hard to see when you're doing it for yourself, but it can be valuable to have an outside point of view. And Juliano and Hayden and I can also help out with that as well. Okay. So let's see, Mike.
All right, yes. So we are finishing up a little bit with the code and such. Last week we kind of divvied up, split up the different assignments that we have yet to do. So I'm working on the NanoHub side of things, getting that uploaded and working. Yeah, I think it's going fairly well.
Okay, great. So let's see. Jonathan.
Yeah, so we met yesterday. We're going to meet again tomorrow. In terms of working on the code, Elmer can get you a bit more insight on that. I know he's been working really, really hard on it the past few days. Meanwhile, JH and I have been working on the write-up for the report, documentation, and getting ducks in a row ready for writing some of the additional requirements. NanoHub, the video presentation we're planning on making. So once we have the simulation, we'll be ready to roll on those.
Great. Elmer, let's jump down to you. Is there anything you want to add to that?
Can you hear me? Hello? Can you hear me?
Yes, yes.
Yes, great. Yeah. Yes, so the code is actually now more or less working. There is one thing, there is this concept motion that I not yet have implemented or tested if it works, but all the rest seems to work. But and we plot the input data because it's so that we do this knockdown, and then the knockdown recovers over time. So we display how for example adhesion becomes stronger, or how something develops from the input parameter, and we still have to make plot from the output, what we get out, so how the result looks like to the cell. It's the input was more important to track down errors now in the beginning, but yeah, more or less we are there with the code, and now the whole rest have to be done. Yeah, and they're already, I mean, Jonathan and GH have already started there a lot. Yeah,
Great. So, Nick, I forgot to ask you if you had anything to add to what Logan was saying.
so thank you very much. as well
No,
yesterday.
he covered most. Oh,
Right.
Sorry, can you hear me?
No, that's fine. Yes.
Yeah, I've been mainly focusing on the Nano Hub submission because as Logan said, we're pretty close to being done with the code. So I heard that can be a long process, so I wanted to get a jump on it. But after that, yeah, just on the report.
Now, once you have the first version uploaded, then updating it is less work, but getting that first version up and distributed is a pain.
So I have like the tool page created already, and I was going to ask Juliano about this, but maybe we can talk in class if there's time.
Yeah, I just got the email from NanoHub saying that I got access. It took them a while.
Oh, awesome.
Yeah, so I just sent you an email, but I recommend you hit the my code is committed, working and ready to be installed ASAP. That's the first step that James was talking about. Then
Awesome.
you'll know if there are bugs to fix and issues with NanoHub that I'm sure Elmar can talk a lot about.
Sure. So I need
I
to
just know the error. I just know that there's an error. I have not yet figured out how to fix it.
but there's no way to test my code until after I hit that button.
Yes, but and importantly, that code, that button will not make your tool public. There's a next step for that. So you can test in private as much as you need still.
It sounds very final, so it was intimidating, but okay. That's
Yeah,
great.
it does, but it's not.
And you can always update the code.
Yes.
So if you find bugs, you can update. So obviously you don't want to publish something that's a disaster. Although goodness knows we've released some releases of Computational that were pretty disastrous. So it happens. Goodness knows Microsoft sometimes releases updates that make a mess.
Windows 8, anybody?
Yes. Windows ME. So these things happen. I mean, if you want to screen share, discuss something, it's fine. I'm not trying to prevent you from doing that if you think that's useful. Otherwise, your choice. I don't mean to rush people. Okay, I guess then we have Carmen and Ibrahim.
I can go. We're also in the same group. But like Mike said, we split up like all the tasks. So me and Hayden have been working on the report and presentation, kind of just finishing things up and putting final touches on it.
Okay, that sounds
Yeah,
good.
I can talk about the code a little bit then, I guess. We are pretty much done, I guess, as much as we're preparing for next week at least. There's just a few values I want to tweak here and there to make sure, I guess, everything looks nice. But yeah, it's looking good. We have been able to introduce cells to the system, and then also slight chemotaxis towards if a cell is outside of its designated area, slight chemotaxis towards like the field that it's attracted to or that it's supposed to be in. Problem I'm having mostly is the cells are not looking very circular. So I think if I just add a surface constraint and just decrease the surface constraint a lot, like make a very small surface, target surface, I can get very circular cells. So I'm going to try that, and we'll see how it goes.
So this was something that I was working on with Elmer over the weekend, which is something that we didn't really discuss. We never played with the surface constraint very much. By default, the surface constraint sets the target to be the same as the area of the cells, which is a little bit odd. So when you're setting the target surface for a cell in 2 dimensions, there's a simple way to think about it, and actually Morpheus does it explicitly this way, which is, suppose that you have a cell that's 7 by 7 voxels, so it's area 49. And I imagine it being a square. What's the perimeter of a square of side 7? It's 7 times plus 7 plus 7 plus 7, 28. And so that 28, a surface of 28, is the smallest surface that you can have possibly for a volume of 49. So if you set the target surface to be 28 for a volume of 49, your cells will basically become square and stiff. They'll freeze. On the other hand, if you set it to be twice that, your cells will get floppy and begin to dissociate. Think about an underinflated beach ball. Basically, you've got a lot of surface compared to the volume. It winds up that a surface that is about 1.3 times the square root of the volume, target volume, works pretty well. If you want the cells to be a little bit more round, you might make it 1.25. If you want them to be a little floppier, you might make it about 1.4. But as a rule of thumb, try taking the square root of the target volume, multiplying by 4, and then multiplying by 1.3. And that will give you a target surface that's pretty close to being what you want for cells that are roundish, but have enough flexibility to move and rearrange. Juliano, do you want to have is there anything to add to that? Now in Morpheus, when cells grow, they automatically readjust that target surface to keep that ratio, that 1.3 ratio. In Computational, if you actually are doing cell growth and division, you have to, by hand, every time you update the volume, you have target volume, you have to update the target surface as well. So you have to do one extra step in Computational compared to Morpheus. You have a little bit more control, fine-grained control that way, but it does mean one more step in your step function. Good question. Okay, anything else that we need to go over right now? Everybody has a neat project, so I'm looking forward to this. Gabriel is not available today, so we'll talk. We'll hear from him next week. Coming back, is the screen share working again? Now I'm going to be asking that repeatedly through this. Okay, reminding us what we did last time. We did this great self-constructed gradient simulation. That was in the lecture 12 demo, self-constructed gradient. And we explored how the trajectories of the particles interact. We explored what happens when the field comes back after some amount of time. What happens when you have multiple cells interacting with each other. And we also learned how to specify chemotaxis in Python, so we had more fine-grained control. And we ran, although we didn't really go into the details, a little demo called Chemotaxis Dependence on Parameters, where we could calculate the velocity of the cell in response to a fixed chemical field as we changed the lambda chemo. And when you do that, you see that there basically are three regimes. When lambda chemo is very small, the cell velocity increases with lambda chemo, but slowly. Then there's a transition to a linear regime where the velocity is essentially linearly proportional to lambda chemo. And then when lambda chemo gets to be very large, the cell velocity is independent of the lambda chemo. And that's because the lattice in Computational creates an effective speed limit. And if you look here, the velocity here is in units of voxels per Monte Carlo step, and you'll see it saturates around 2.24. So the fastest a cell can move is about 2.24 voxels per Monte Carlo step. In practice, you want to stay below that. And so you should look for 0.2 voxels per Monte Carlo step or a little bit less. And so when people ask the question, what does Monte Carlo step represent in real time? One way to figure that out is you can make a Monte Carlo step represent as small a time as you like, but on the large side, you can't have a Monte Carlo step be bigger than the amount of time it takes for a cell to move about 0.2 voxels. So if you look at your simulation, you find the objects that are moving the fastest and ask the question, how do I set a Monte Carlo step so they don't move more than about 1/5 of a voxel per Monte Carlo step? That sets a maximum upper limit on the amount of time that a Monte Carlo step can represent. And that simulation is designed so that if you wanted to scan something else, like the velocity of the cell versus the target volume, or the velocity of the cell versus lambda volume, lambda surface, you can easily change that simulation to study the dependence of movement on all of these other parameters as well. And we did play a little bit with the idea of adaptation. These days, the Chemotaxis plugin has specifically a log scaling coefficient that allows the force on the cell to be independent of the concentration. That's proportional to the gradient divided by the concentration of the cell. And that allows you to get the velocity of the cell to be linear. That is, the velocity to be constant, actually. I should be careful. So the movement of the cell is linear, the velocity is constant, even though the chemical field is exponentially changing. If you do a linear chemotaxis in an exponential field, the cell moves very, very, very slowly at first and then accelerates rapidly at the end. In biology, the cells will essentially crawl at a more or less constant rate, independent of the concentration, the mean concentration in the gradient. Okay. So a couple of things that you might want to think about. We haven't done a lot of principles of modeling in this semester. We've been focusing on code, but it's worth coming back perhaps when we think about chemicals and ask the question, if you were looking at a biological problem, how would you begin to translate that into a model? Now, most of the exercises people are doing are replicating existing models. So somebody's already gone through this process. But for example, Elmer wanted to rebuild the simulations from the paper he was looking at, and JH and others, rather than to just directly translate the Morpheus code into Computational. And so this kind of thinking about the biology and thinking about how to translate the biology into models, I think, is something that is worth thinking about. And chemotaxis is a pretty good place to think about this. A basic thing to think about is a lot of biology is about signals and responses. And so if you have a signal, the first question you ask is who's sending the signal? Second question is who's receiving the signal? And the third question is how does the cell that's receiving the signal change its behavior in response to the signal? And so if we're thinking about chemicals and cell responses, to begin with, you might ask the question, where is the chemical? Is it diffusing freely everywhere? Is it diffusing freely in the extracellular space? Is it bound to the extracellular matrix the way it was in our self-constructed gradient example? Can it cross the cell membrane or not? Is it attached to a substrate in some way? Or is it in 3 dimensions? When cells and tissues move, did they change the chemical field? In reality, they usually do. If I'm pushing through a fluid, I move the fluid. CompuCell, we don't usually simulate that. So you have to ask the question, is that neglecting that important or not? Another thing that can be quite important, which we talked about a bit, is that when a cell receives a chemical signal, is that signal removed from the environment or not? If the concentration of the molecule is very large, and the cell's only taking up a little bit of it to sense it, then that may not be so important. But we've done plenty of simulations where the cells are taking up oxygen or glucose, a substantial fraction of what's available. In that case, the cell sensing itself is changing the environment, chemical environment. In that case, we need to know how much we're taking up. On the side of the receiving cell, we have to ask the question, how does the cell know what the environment is? Are the receptors for the signal all over the surface of the cell? Are they only on some surfaces? Are they only active if they're in contact with a particular thing? For example, endothelial cells that we worked with last week often will only sense vascular endothelial growth factor if it's actually bound to the extracellular matrix. What is it that the cells are actually sensing? The direction of the highest concentration, the direction of steepest change, or something else. When people think about ants and ants following pheromone trails, as you know, when ants move, they lay down a chemical trail. Other ants can detect it. If you do a naive simulation where you say that the ants move towards the highest concentration of the pheromone, what you find out is you have a big pile of ants that don't move. It's a little bit like the coalescent simulation that we did for endothelial cells. And so you have to think about what the response is to that signal. We already talked about how the number of receptors changes in response to the concentration. That was our logarithmic adaptation. But the location of the sensors on the cell could change too. Sometimes the sensors start out being dispersed over the cell and then localized, in which case the directionality of the cell movement becomes more pronounced in time. When you receive a signal, typically that blocks the signal from being received again over a short period of time. And a lot of things can happen in a biological sense. Quite often, the molecule that receives the signal is internalized into the cell after the signal is received. And it may be broken down and need to be resynthesized, in which case the sensitivity of the cell goes down to the signal. Sometimes it's recycled back to the membrane. Other times, the molecule binds, sends its signal, and then unbinds from the cell, in which case the receptor can stay on the membrane throughout the process. And then you have to ask, what is the signal inside the cell? How does that signal, whatever it is, depend on the fraction of bound receptors? Is that signal level per bound receptor the same, or does it change? Is there adaptation there? And is that signal a one-time thing where when the molecule binds, it sends a pulse and that's it? Or is the signal active as long as the receptor is bound? Those are all important things to think about. A lot of those require the modeling of network responses, biochemical reactions and network responses that we'll talk about a little bit later today. And then what does the cell do? Does the cell move in response to the chemical only, or does it move even when there's no chemical? Is there some minimum level of signal below which the cell ignores the signal? Does the cell respond as a whole, as if it were single agent, or do different parts of the cell respond independently to each other? For example, in neuronal outgrowth, in axonal outgrowth, each growth cone of the axon will follow chemical trajectories independently. The cell as a whole may actually, the soma of the cell around the nucleus may not move at all, but the membrane may project and build branch structures. In other cases, the cell behaves as a unit. And it comes back again, is every surface of the cell responsive or not? Is the velocity of the cell independent of the gradient or independent of the concentration? Is the movement of the cell deterministic or moving through biased random walk? We didn't go over in class the difference between chemotaxis and chemokinesis, but there's a set of slides that compare how eukaryotic cells move in response to chemicals to the way bacteria move. How do cells combine multiple potentially conflicting signals? And in the case of things like dictyostelium, which we saw a video of, dictyostelium move in a pulsatile way. For a constant concentration of chemical, they'll typically move, stop, move, stop, move, stop. And so those are some of the kinds of questions we might ask when we start looking at the biology and want to translate that into a simulation. We then looked at vasculogenesis in response to chemotaxis and did a little demonstration of that. And the next thing that we would normally do, and I'm going to skip this because of time this year, is there's a very famous movie from the 50s of an immune cell here moving through, this is in blood, the non-moving or barely moving circular and spiky things are red blood cells. Here we have a macrophage. Those two little dots are bacteria. And this is an immune cell chasing and eventually eating a bacterium. And the immune cell is responding to chemicals that are secreted by the bacteria in the environment. And there's a demo that's packaged with CompuCell called Bacterium Macrophage, new style in this case. And that's a not very pretty simulation that is a gross caricature of what's happening in this simulation to the left. I mean, this experiment to the left. And one of the exercises that we often do in class or in homework is to take that very simplified simulation and modify it bit by bit to make it more similar to the movie. And here on the right, I have a more advanced simulation in the online, in the packaged model. The red blood cells are just fixed; they're barriers. They don't respond. Here you see a variety of simulated immune cells in blue chasing bacteria in green around the obstacles provided by the red blood cells. So now I want to come to persistent cell movement. Cells move, in our world, cells move in response to gradients of energy. In reality, If I park a car on a hillside and take the brake off and put it in neutral, the cell will roll down the hill in response to the gravitational potential energy. But most of the time when I'm driving, the movement of this car is not dependent on the external potentials acting on the car. They may slow it down a little bit or speed it up. But the movement of the car is dependent on the motor inside the car. And similarly, when cells move, they have rather elaborate molecular motors inside of the cell that allow them to move. And they have a variety of motors that they use. These have a variety of names based essentially on their shape. Filopodia, which are essentially fibers, which the cells extend, anchor, and then pull on. So it's a little bit like taking a rope with a hook on the end of it. The cell throws the rope out. If the hook hooks onto something, the cell then pulls on the rope and pulls itself towards the bounded spot. Lamellipodia, where the cell has a sheet-like lamellar structure, which it extends over usually flat surfaces. And then ruffles, which are the three-dimensional equivalent, or what's called the amoeboid moment. All of these structures are built using actin and myosin. Actin is a polymer, complex polymer, and the construction and destruction of filopodia, lamellipodia, or ruffles takes time. Typically, the lifetime of any of these structures is a minimum of about 30 seconds, a maximum of several minutes. In some cases, they're persistent for very long amounts of time, but typical time scales are about 30 seconds to a few minutes. And so that means that once a cell starts moving in a given direction, it tends to continue to move in that direction for some amount of time. Now, if your Monte Carlo steps are long, representing 10 minutes, then the direction at one Monte Carlo step and the next Monte Carlo step may be relatively uncorrelated. But if your Monte Carlo step represents 5 or 10 seconds, then there'll be a strong correlation between the direction a cell moves in at one time step and the next time step. And that correlation of direction is known as the persistence of the direction. And that's why this is called persistent cell movement or persistent migration. Computational cells by themselves, due to membrane fluctuations, move randomly with no persistence time. And so if we want the cell to move persistently, we have to impose that by biasing cell movement. And Computational has a plugin with the not very edifying name of LambdaVec, which allows us to apply an external force to the cell. And we can use that force to then generate persistent movement. And that's what we'll do in class now. So the first exercise is to create a cell with an external force on it. And I'll walk through the exercise first, just outline it with you, and then we can go over it again. I don't know, Juliano, if you want to walk through it again, or maybe I can do a split screen and do it maybe that way, maybe the most effective, so that you can see what we're going to do. Almost everything we need is in Wizard. We'll have to do a little bit of editing of our code. And if people get stuck, under Lecture 13 demos, there's a simulation called single cell persistent movement that has the finished code that we're going to develop. So the first thing we're going to do, and if you want to read about external potential, it's in the manuals under external potential plugin. We're going to create a 256 by 256 by 1 lattice. We'll have it have periodic boundary conditions. We'll use blob initializer to create a single cell. And we're going to set the pixel copy range to 3 to begin with. Okay. Then we're going to skip no chemical fields in the simulation. And we have to give ourselves a volume, so we can say that there's, we'll have a contact energy of the cell. So for contact, we will give the cells an individually defined volume, volume local flex, although we could perfectly well use volume flex for this. We're not going to be playing with that in this particular simulation today. If we were going to go further, we'd need that. We will click external potential local flex. That's our new feature. And then we'll use center of mass and cell neighbors as well. As usual in Computational, you can set the external potential in the XML. But in this case, if you apply the same force to every cell, it's sort of boring. That would be like putting a gravitational potential on everything. So typically, we want to be able to vary it on a cell-by-cell basis. The one thing that we're going to keep from the external potential is this algorithm tag. And there is what's called a pixel-based or a center mass-based formalism. This comes back to the idea of where the force is acting. Is it acting on each point of the surface separately, or is it pushing the whole cell as a unit? If the cell is a compact round object, this won't matter. But if I had a very complex cell shape, this might matter quite a bit. We're going to use pixel-based, but you might want to try the other one, see what it does. Then we're going to change the blob initializer to create a single cell. And then, of course, in the Python, since we used volume local flex, we have to assign the cell a volume. Here, we'll use target volume of 65 and lambda volume of 8. And then the next thing that we're going to have to do is we're going to need some random numbers. Computational, the current release, loads NumPy as NP by default. If you're using an older version of Computational, you may have to load NumPy as NP to have access to that library. And NumPy has a variety of random number generators, rather too many. The ones we'll use are np.random.random, which takes no argument, which simply generates a uniform random number between 0 and 1. And np.random.uniform, which takes as an argument min and max and generates a uniform random number between min and max. So if you want a random number between -1 and 1, we'd say np.random.uniform parenthesis -1.0 comma 1.0. And we can generate Gaussians and Poisson distributions and other things. That's all in the NumPy manual. Again, we're not going to need all of that detail today. And then we want to apply a random force to our cell. And what we really want is a force that's constant in magnitude, but it's going to change direction. And the force is specified as a vector. And having the vector change directly is a little bit inconvenient. So what we'll do is we'll define the angle at which we're applying the force. So here, cell.dict angle is 2 times pi times a random number between 0 and 1. That gives us a random angle. And then we'll convert that angle into the x and y vector components of that vector, NumPy cosine and NumPy sine. For the moment, we're not going to use the persistence time, but we'll just define it because we'll need it in a few minutes. And so we'll define a persistence time for the force, which we'll call alpha. And we need a strength for the force, called propulsive force. Now that we've defined a vector to put onto our cell, we can apply it to the cell. Cell.lambda vec x puts a force in the x direction, and we'll make that equal to propulsive force times that dir x that we calculated. And as usual with Computational, higher energy is against us, so we need a minus sign to push in the direction of the vector. We'll do that in Y, and then we're in 2D, so there's no Z. And we should see that the cell moves. And in Twit, I never remember where it's hiding. Juliano pointed out to me that it's under cell constraints, apply force to cell, if you don't remember how to do this. Okay. And then it's useful to apply, to plot a center of mass position for the cell. And so here we'll create two windows, one of which will plot the X and Y position of the cell versus time, and one of which just plots the position XY of the cell, so we can keep track of where the cell moved. Okay. Now we put all of that together, and we get a cell that will move. So why are do people want me to build this in front of you, or would you like to try working on it on your own for a few minutes? I've got all the key code elements here on the screen so that you don't have to thumb back to the other slides. What do you think? What would be most useful for you? So let me show you how I would go through this. And I'm going to go back to the beginning of our exercise here. And I'll do a split screen share. Make sure, does that look okay to people? You have Twitted on the left, and so what we're going to do is we're going to go to CC3D project. We'll do new CC3D project. And I'm going to call this in-class demo one, because I already have the one that I want, the name I want. And I'm going to make the system have periodic boundary conditions. I'll move the pixel copy range to 3, and I'll change blob initializer. Okay. Do people want to follow along, or is it okay if I keep going? If you want to follow along with me, that's fine. Let me wait. Give me a thumbs up if you're caught up with this. Okay. So now I only have one cell type, so I can just call it cell. Add it. I have no chemical fields. I skipped that. So everybody get up to there. Elmer says, okay. Everybody all right up to there. And now, good. Okay, and now I'm going to say contact. Volume local flex. External potential local flex. And that's it. So go ahead, we'll do that. And we now have some code. And so the first thing I'm going to do is go.
James, is it possible that you make the 3D a little bit wider and the presentation a bit slower? That gives a little, then it's better readable. No. Or maybe it's just my screen that, yeah.
All right, thank you.
It's okay, thank you, thank you.
How's that? I can probably change the font size here, be a little bit bigger. Let's see.
The move maybe be great. It's quite pixelated on my screen. I don't know how it's on other screens. I just know my screen.
How's that?
Yeah, that's much better. It's not perfect, but much better.
Okay. Anybody else having trouble seeing? All right, so now in the XML, the only thing that we're going to have to do is start with a single cell. So instead of a radius of 51, we'll use a radius of 3. That's all we have to do. So we'll hit save. And now we'll go to the Python. And the Python is the place that we have to make some changes. So the first thing we have to do is we have to import NumPy as NP, if you don't have that. I just upgraded my CompuCell to 4.4.0. So if you're using an older version, NumPy may not be there, so you have to put that in. And now we are going to, in our start function, we are going to have to create the properties of our cell. And so we will go to CC3D Python, visit all cells. Get rid of this text that I don't need, so it's easier to see. And now what am I going to do? I'm going to have to first create the target volume and target surface of the cell. So cell dot target volume. I think I said it was supposed to be 65. Cell dot lambda volume equals 8. And now I have to start building the things that I'm going to need. So I need to have an angle for the cell. I'm going to create a dictionary entry for the angle. And this is going to be a random number in radians. So I multiplied by 2 times NP. NumPy has pi built in, so NP times pi. NP.pi. And then
Just a question, is this in the start or is this in the step level that we have?
It's in the start function.
This is still in start. Okay, sorry.
You have to give your cells properties or they'll disappear. You don't put them in the start function and the cell will disappear. And so now I'm going to use the random. And one thing that's always a bit funny in NumPy random is it's random.random because there are multiple randoms. And so we'll do random.random. That's one where you have to look it up if you don't remember. And now I have a random angle. And now I want to get the x coordinate of that random angle. And so I'll call that direction x. And I'll set that that is equal to NumPy cosine of angle. Oops. Easy to make a mistake there. NumPy of cosine of that angle. And I'll make the same thing for y. Sine of the angle. Okay, give me a thumbs up if you can keep up with that. If you want me to go over it again, that's fine. Anybody? Ibrahim says that's okay. And now I'm going to define the amplitude of my force. So I'll say cell.dict. And you could give these things shorter names, save you typing if you want. Cell.dict, repulsive, oops, I spelled it wrong. Force. And that is going to be the force amplitude, and there's no particular reason that it should be 20, but I happen to know 20 works okay. 40 might work better. 20 is reasonable to start with. And I'm going to define this persistence time, which I'm not going to use right away. I should probably put that in a little bit later, but we'll call this alpha. And I'll just set that equal to 0.99. Okay. And I'll wait for a minute to make sure everybody's caught up. So give me a thumbs up when you've got that. I'll point out that so far we haven't done anything except create some variables that belong to the cell. We haven't applied any force to the cell. And if you need it, I will actually try. This won't work if you're using NanoHub Computational. It'll work on desktop. I'm going to put the code in the chat if you like. Just type. Okay. And so the next thing that we have to do is apply the force to the cell. And if I don't remember how to do that, we go to cell constraints, apply force to cell. And now the force on lambda vec x will be minus repulsive force times the direction x. And for y, I want the same thing, except I want the y direction. Okay. Why don't people try that out? And why doesn't somebody run it and tell me what happens? I'll call on somebody. Logan, what happens when you run that?
Sorry, my computer's being slow. I'm still.
Mike, is that working for you? How about Ibrahim or Nick?
I
For
mean,
me-
I go to,
Oh,
go ahead.
you got me. I can, it just pushes it in the same direction for the entirety of the simulation.
Exactly. Each time you run it, it should go in a different direction though, right?
Yeah.
Okay.
Run it again.
You want to show us? We can take a quick look.
Size my windows. Do you want to see? I mean, I guess my code is probably uninteresting. So let me just show my simulation.
Well, if you did the coding a different way, that could be interesting, right? This is pretty.
No, I just followed along.
Yeah, there you go. And so you've also added the plotting of the position as well.
Mhm. So now it's going this way.
So one thing you might want to do is why don't you stop that and make the force twice as big. So make that um,
20 to 40.
Yeah.
Try it. It was definitely going faster before I was on Zoom, but I'm afraid this always happens. It's a bit better. Huh, and it picked a pretty similar angle this time. See if I stop it and restart it, we'll pick a different one. Okay, yeah.
Well,
I'll
actually,
it downwards.
okay, great. That looks good. Anybody else get that working or having trouble getting it working? Good. So we can add some plots. That one I don't want to take too much time on. Just remind you that we can add a plot here if we want. Again, CC3D Python, scientific plots, setup gives us most of what we need. We're going to plot the center of mass. I'm not going to type it out as nicely as I did before versus time. So this will be position. And we'll define, I called it here XCOM, YCOM. I'll change the line to style to be lines, color green, and size 5. That will give us a plot of the X position versus time, the Y position versus. And then it's nice to have a history of everybody the cell went. So I can copy this and create a second plot window, plot win 2. This will be something like cell position. And in this case, the x-axis is just X position. The y-axis is Y position. And now the series that I'm going to add, I have to change this to plot win 2. Call this position. And again, style of lines. Maybe since those are green and red, I can make it blue. I don't need the second line. So that creates my 2 frames. And then in the step function now, I'm going to go over the cells in self.cell list. And now I want to do plot, add my points, SPN scientific plots, add data point. And for the plot win one, I have to remember what I typed. The name of this was XCOM. And now I have to remember it's cell dot X capital COM as the center of mass in the X direction. YCOM, cell dot YCOM. And now for plot window 2, instead of plotting the positions plot window 2, instead of plotting, what did I call it? I have to remember. I called it position. It would be nice if you didn't have to remember what you typed. And now I want cell.xcom versus cell.ycom. So let me give people a minute to try to get that working. Again, you don't have to do too much. Almost everything you need is in the Twidit menus. if you like, I can give you the code here. That's to initialize the plots. I don't need to print the cell ID anymore. So get rid of that. Save some space, make it easier to see. And here's the plot. If somebody wants to try that and see whether it works, quite capable of making a typo, so people should check. And that should give the result that Nick just showed. Okay, people need more time, or is that working for you? Last call on needing more time. Elmer says it works. Anybody else? If you don't want to speak up, you can always put the request for more time in the chat. Don't be shy. Okay. So we ran this now. And what we got was what Nick showed to us. You'll get the cell moving in a single direction. You'll notice there's some fluctuations because the cell does have some random movement to it. But it's a pretty boring simulation. The cell will just keep going in a direction around and around and around. And if you have set things up right, each time you run it, the cell will move in a given direction. There's some noise. And that noise is because of that intrinsic stochasticity of the Potts methodology. And if you change the propulsive force, you'll find that as you make the propulsive force bigger, the cell moves faster. If you make the propulsive force too big, the cell will either break or just move at a fixed velocity, just like for chemotaxis. Okay. So here is the next exercise for you. Now, in the step function, what I'd like you to do is update the force. And the way I want you to do this is we are going to imagine that the angle that we start with is going to gradually drift randomly. And we could do that in a lot of different ways. But what I would suggest as a simple case would be I take the angle that I have. At each time step, I make that angle what I had before. And then I add to it 2 pi times a random number between -1 and 1 times 1 minus that alpha. So if alpha were 0, I'd have a completely random new angle every time. If alpha were 1, my angle would be constant. I wouldn't change anything. And then I have to recalculate, once I have the angle, I have to recalculate the things I did at the beginning. I have to recalculate dir x and dir y. And then I have to redo lambda vec x and lambda vec y. And you can literally cut and paste all of the code from lines 30 down to 37. You won't need lines 33 and 34 because you're not changing those. And then you'll have to update your paste in of line 30 so that instead of having a random angle, you now have the old angle plus a random number. Okay, is that clear? So why don't people take a couple of minutes and try that? And this is a little bit more interesting a simulation. Now you could pick a lot of different ways for the angle to shift in time. But here you have something rather simple, which is, again, if alpha is 0, then the angle is randomized at every time step. If alpha is 1, the angle is totally persistent. And if alpha is between 0 and 1, the bigger alpha is, the longer the persistence time of the cell is. And you should be able to see that very clearly when you run the simulation. So why don't you try that? And you can play with alpha and see what happens between those two limits. If you've got that done and you want to go a little bit further, it's interesting to plot the angle of the cell versus time, just as an extra plot, create a third plot window. And the code for this is, if you get frustrated, single cell persistent movement V2. Okay. Any questions about that? So why don't people take just a few minutes and try to add that code to the simulation. Again, you can cut and paste lines 30, 31, 32, 36, and 37. And then all you have to do is edit the pasted version of line 30 so that you're keeping track not of the changing the angle directly, but incrementing it or decrementing it. And that's why I use this random uniform to pick a random number between -1 and 1, because I want the angle to be able to go up or down. Okay, let me just walk through this with you. I'm going to copy lines 31, 32, and 33 and put them here. And then that will calculate my new angle. And then I'm going to copy lines 36 and 37. And put them here. And I have to get my indentation right. And the one thing that I'm going to have to do is change the angle. And I'm adding something to cell dict. So instead of retyping cell dict angle, I'm just to do a plus equals here. And now I want n pi, pi, np. Instead of using random.random, I want a number between -1 and 1. So I'll use uniform. -1.0, comma 1.0. And if I did that, it would be basically wiping out my angle at every time. So I'm going to multiply by 1.0 minus cell dict of alpha. Okay. Let's see what happens if I try to run it. No guarantees. So let's see, open and flag. My cat has decided to join the class. Let's see. Huh. Okay, what happened? Oh, there it is. Just slow. Just slow. If anybody has it working faster than I do, please speak up and we'll demo it. We'll have you demo. Elmer, is it working for you?
No, it's not working yet. I got somewhere stuck and lost.
Sorry?
It's not working. I got think that it's working. I copied and it's something wrong. So I'll try to figure out what it is.
Anybody else? Is it working for people? I may have a typo. I may have, let me see if I can open this player. Oops, oh, that didn't work. So I just had Twitter die on me. So let me try again. It worked correctly when I ran it earlier today. It may not like the Zoom share. If people are stuck, there's the code online in that single self-persistent movement group too. Okay. Okay, so this is what happens when I run that code. I'll restart it. And this version, I actually have the cell angle plotted as well. There's some strange interaction between player and zoom so that the axis labels are messed up. Not sure where that comes from. That seems to be a resizing issue of player. But here you see the cell position. You'll see the cell moves generally in a continuous direction, but over time it loses its memory of what direction it was moving. Does anybody else get something similar? Does anybody need help with this? If you're having it not work, we can debug it together. So why don't somebody volunteer and we can go through. Ibrahim, how's it going?
Yeah, I got it to work.
You've got it working. Do you want to show it?
You want me to show? Yeah, okay.
Sure.
Can you see?
Mm-hmm. Yeah, so that looks pretty good. You don't have that bug with the labeling of the axes. I think the reason I was getting the axis labeling was that I opened it on my laptop and then I moved the player to the screen share I use for Zoom. I didn't resize properly. So that looks great. Anybody else? Is this working, not working for people? Anybody need help with it? Okay, so I hope people will play with that a little bit. Changing the persistence time is interesting. So let's do that one thing before we break on this. Let me just run. Let me do a split screen again. Hope this will work. Okay, and here I was using an alpha of 0.95. So let me try first an alpha of 1.0. And I'll hit it. You can see the code, right, at the moment. And now I'm going to play this, go to player, and I'm going to restart. Can people see the player all right? Okay, I'm going to restart it. And with the value of alpha 1.0, the angle is fixed. So the cell moves in a single direction with a little bit of noise. Okay. I'm now going to go back and make the persistence of the force 0. So now the cell should move totally randomly. And let's see what happens. Well, I see the cell position is basically moving around randomly. Looks like the angle, I bet I know what's happening here. Looks like the angle is actually increasing. And I think the version of this code that I have may not be quite what I wanted. It may not be quite what I have on the screen here. Where I calculated the new angle. No, that should be correct. Yeah, that should be correct. And let me try an alpha of 0.95. So that should have a moderate persistence time. Takes a little while to see what's going on, but the cell is going in a given direction for a while and then turning around. If you look at the cell angle, you'll see the way that changes. I'm actually a little puzzled by why it seems to continue to increase, because it should be uniform, random, up or down. Okay. And you can get a sense of the persistence time here. It keeps going in the same direction over a period of about 20, 30 Monte Carlo steps. If I make the persistence time longer, like 0.99, see. That is non-pi uniform random, so it should be correct. 0.99. Let's try that. Now it should stay in the same direction for longer. You see the velocities in the X and the Y direction are almost constant. They've got a little bit of fluctuation. And if I wait long enough, I'll lose the memory of my direction. So that could be a nice exercise for people to play with. You do have to wait a bit of time for it to show its behavior, but you should be able to get something that looks like that, where the cell looks like a string, a coiled string. The trajectory of the cell looks like a coiled string. Were people able to get something similar? Did it work, not work for people? Anybody have any comments? There's actually a lot you can do with the demo like this. It's a simple but relatively powerful way of getting cells to move. And if you actually know the distribution of angles and the persistence time, you can impose those from experiment on the cell. Okay. I want to come back to one more thing. And again, this is an exercise that one could do in class. This time, I don't think we're going to take the time to do it. Once we have a single cell that moves persistently, we can begin to build tissues that move persistently. And the trick to building a persistent tissue movement is to say that the updating of the force on the cell is not random, but depends on the direction that the cell moved at the previous time step. And if you do that, you can actually get the cells to move the way cells move in healing tissue, where the cells at the boundary of the wound begin to move in. And they send a signal that causes the cells right behind them to move in the same direction. And that causes a signal to the cells behind them to move in that direction. The net result is that all the cells move towards the wound, and the wound closes pretty quickly, which is the simulation you see down here. And that simulation, which we're not going to run today, is available to you in lecture 13 demos per day. The T is missing. It's persistent motion that does this. Why is this important? Well, if only the cells at the boundary moved and they had to pull all of the tissue behind them, it would be like a coal train where you have a locomotive that's pulling 200 carriages full of coal. And as we know, one of the things about coal trains is that they take a long time to start moving. They don't move terribly fast, and they're hard to stop. If we want fast acceleration and deceleration in a train, we do what a subway car does or a commuter rail car does, where every car has its own motor. And the car in the front starts moving, it sends a signal to the car behind it to start moving, and the regulation controls the spacing between the cars. And that allows very rapid acceleration and deceleration. For passenger service, where the trains stop and start a lot, that is definitively different in terms of the efficacy of the train. And so that's really all I wanted to show about explicit forces for the moment. I'd like to now move to talking about networks. So if you took my course last semester, you'll know what we're going to talk about. If you didn't take the course last semester, you could take it in the fall, and this may go by a little bit fast. So if it's a bit unfamiliar, think of this as a quick tasting of how these things work. We're going to be talking about subcellular networks primarily. And the reason we're going to be focusing on networks is that networks are active in a variety of scales in biology. In particular, at the whole body scale, blood circulates chemicals in our bodies, which behaves like a transport network. Within cells, we have biochemical reactions, and for example, in metabolic networks, and we have other kinds of transport and reactions that we're going to talk about as well. There are a whole variety of resources that we have specifically for this. The fall course is taught out of a textbook by Herbert Sauro called Systems Biology Introduction to Pathway Modeling, which I like because it's very simple. There's also a nice online quick start manual, which I recommend people have available to them, and I'll put the link in the chat here. So if you want to have an online manual, I recommend looking at the Read the Docs for Tellurium, which is here. Put it in the chat. I also sent around a one-page summary of Antimony, which is the language we're going to be using in conjunction with Tellurium. It's very, very simple code language to write network models in. We can cover the entire syntax in 4 slides, which is why I like it. If you want to understand the details of network modeling, you can get pretty deep, and there are all sorts of great books that you can use for that. There are 4 main kinds of biological network. These are chemical reaction and metabolic networks that create and destroy chemical components in cells. There are signaling networks that transfer networks from outside cells to inside, transfer information from outside cells to inside cells and within cells. And there are gene regulatory networks that turn genes on and off inside of cells. Now, gene regulatory networks are a little more complicated than they're led on to be, because in addition to things like transcription factors, you have things like methylation and demethylation of DNA and methylation, acetylation, and their inverses of histones that regulate gene transcription that are not usually thought about as part of the regular GRN. There also are small interfering RNAs that are not normally thought of as part of this gene regulatory networks that are critical as well. And then we have transport networks, which go under the somewhat ungainly name of physiologically based pharmacokinetic networks, mainly because these come from the pharmaceutical industry trying to understand how drugs are distributed in the body. Chemical reactions are typically written in the form of arrow diagrams. All of these are going to be written in the form of arrow diagrams. We can have here, for example, a classic chemical reaction. 2 molecules of ADP go reversibly to a single molecule of ATP. Adenosine diphosphate goes to a molecule of adenosine triphosphate and a molecule of adenosine monophosphate. Effectively, what we've done is move one phosphate from one of the molecules to the other. And that phosphorylation reaction is a key thing that happens in biological signaling. Classically, we'll have multiple chemical reactions here. 2 molecules of A go to B. B goes to 3 molecules of C. C plus A goes to D, and so on. The nodes, the things that are being changed, represent the concentrations of chemical species, and the arrows represent chemical transformation. Every arrow has implicitly a rate, which is the rate at which A is converted to B. And that's the fundamental concept of dynamics on networks, that every arrow has a rate associated with it. Chemical reaction networks conserve mass And in general, metabolic reaction rates are faster than the other things we're going to be modeling. If we're going to talk about chemical reactions, there's one fundamental place to start with chemical reactions, which is the conversion of an arrow diagram like A plus B goes to C into a set of ordinary differential equations. If A and B combine to make C, then clearly the rate at which I produce C has to depend on the concentrations of A and B. And that rate law, VF of A comma B, means that this kind of mathematics is known as reaction kinetics. Kinetics, rates of chemical reaction. The single arrow diagram, A plus B goes to C, implies that there's a rate of change of the concentration of A, a rate of change of the concentration of B, and a rate of change of the concentration of C, all governed by the rate law that sits on that arrow. In this case, A is destroyed and B is destroyed and C is created. And for every molecule of A and B I destroy, I create one molecule of C. To go any further, we need to have a hypothesis about what the form of that rate law is. And for chemical reactions, the simplest form that we run into is what's called the law of mass action, which says that for a single reaction, A plus B goes to C, the rate is a constant, K, times the concentration of A times the concentration of B, in which case the rate equations become dA dt is equal to minus KAB, dB dt is minus KAB, dC dt is KAB. Why would you guess, besides having taken a chemistry course, that the rate ought to be K times A times B? Well, if I have no A, what is the rate at which A and B combine? It better be zero.
Zero.
No. Similarly, if I have no B, the rate at which they combine better be zero. And so I need a function that's zero when A is zero or B is zero. In other words, it's non-zero when A and B are both non-zero. And the simplest functional form that has that behavior is K times A times B. I can write more complex functional forms. In particular, if I have a lot of A or a lot of B, I might have the rate be slower than this would predict. But for small concentrations in normal chemical reactions, you always obey the law of mass action. And so that's always your starting hypothesis for chemical reactions. Things are a little bit more complicated. If I have 2A plus B goes to C, then the law of mass action says K A squared times B. And the reason for this is derived from the probability of two particles interacting with each other. If I have a box and I ask what's the probability that molecule A and molecule B collide, that probability is proportional to the concentration of A and the concentration of B. If I need two molecules of A and one molecule of B to come together, then the probability is A times B, or A squared times B. Protein signaling networks describe the flow of information and control in a cell. And this is typically how cells sense outside world. A classic example here would be a receptor, in this case, Notch. Notch binds to its ligand, which is called delta. The intracellular domain of Notch actually is released into the cell, and then it sends a signal which causes the phosphorylation of a molecule called MAP kinase. phosphorylated forms are often shown with a star. The phosphorylated MAP kinase, phosphorylates MAP kinase. The phosphorylated MAP kinase phosphorylates MAP kinase. And the phosphorylated MAP kinase controls a bunch of additional signals that lead to things like cell proliferation and motility. So a typical pattern of protein signaling is that you have something that attaches or removes a phosphate group, that is, it phosphorylates or dephosphorylates something else. A molecule that attaches a phosphate group to another molecule is called a kinase. A molecule that removes a phosphate group from another molecule is called a phosphatase. The process of attaching a phosphate is called phosphorylation. The process of removing it is called dephosphorylation. And you'll see this kind of cascade of phosphorylation in kinases again and again in different forms if you look at signaling. Why do you have a stack? Why don't you just have the signal immediately go to this output? This kind of stack allows intermediate levels of control where some other signal can amplify or interfere with the signal coming from above. It also allows for very sharp transitions, where as I change the signal at the top, the output goes, instead of from being a smooth mass action-like behavior, to being a step function. There also is typically more resistance to noise in systems of this kind. And so you'll see this kind of cascade happen multiple ways. Sometimes you need multiple phosphorylations of the same molecule to activate it. Sometimes phosphorylation will turn a molecule off and get inactive. There are a whole variety of these kinds of behavior. Gene regulatory networks typically involve the turning on or turning off of the transcription of genes. There's a particular notation of these, which is a little bit misleading because it makes them look very simple. It makes it look like the circuits that you use when you do Boolean logic. In fact, the behavior of genes is nowhere near as regular as this kind of diagram makes out. The little diagram here, which is aligned with an arrow coming out of the end, is meant to represent the DNA on the bottom and the messenger RNA transcript, or the pre-messenger RNA transcript coming off of that gene. And so these kinds of diagrams represent the control of the transcription of particular genes. And what you'll see, even in this very zoomed out view, is that very often the products of genes act to either promote or inhibit the production of messenger RNA for other genes. And you very often have multiple signals controlling the same gene. And those multiple inputs controlling the same output make life rather complicated when you do this kind of model. Gene regulatory networks typically operate on a pretty long time scale, hours, days. Signaling networks typically operate on a time scale of minutes. And chemical reaction networks typically operate on very short time scales, seconds, even milliseconds. Gene regulatory networks often control long-time behaviors like cell differentiation or transition between a cell cycling and non-cycling. Again, there's a little bit of under the hood magic here, because things like cell differentiation can be initiated by gene regulatory networks, but can't be maintained by them. We're not going to go into that in this class. Notationally, a gene is shown as a horizontal line. This arrow coming off of it represents the production of messenger RNA. And there's an assumption that that messenger RNA is then translated into protein. In reality, there are a bunch of additional steps. Very often we'll short circuit this diagram and say that if an inducer binds to the promoter site of the gene, the result is the production of the protein. In reality, if we look at the fundamental dogma of biology, molecular biology, we'll see that there are multiple steps. First, the promoter has to bind to the DNA. The DNA has to unwind. The transcription complex has to bind to the DNA. It produces RNA. That initial RNA needs to be edited to get rid of introns. It also has to have a poly A tail attached to it. It has to be exported from the nucleus. It has to find its way to a ribosome. The ribosome then has to translate the RNA into a raw protein. And then the protein itself has to be folded properly and very often has post-translational modifications. And so actually, this idea that initiator produces protein is skipping over a whole bunch of steps, any one of which can take time and which can be regulated. It's a bit amazing that these kinds of models work at all, but they actually work better than they should, perhaps, in terms of describing the biology. One of the things that we'll see in both signaling and gene regulatory networks is something called motifs. We have the idea of a molecule promoting the transcription of a gene, a molecule inhibiting the transcription of a gene, two molecules coming in. In this case, they're both inhibitory. A gene producing something that then turns off another gene. A gene producing something that turns itself off, autoregulation. The effect of something being turned on, being inhibited by something else, and the regulation of phosphorylation. This molecule can only turn the gene on if it's phosphorylated. And this kind of circuit exists again and again, and there are a whole bunch of these motifs, some of which you see repeatedly in biology. The last thing, that kind of network that we deal with on a typical model, is a transport network. Again, the main concept here is that blood is circulating through our bodies. It's carrying a bunch of chemical species around with it, oxygen, glucose, drugs that we've taken. And that means that molecules are continually being introduced into our organs and taken away again. And we often want to know, if we swallow a pill, what's the concentration of that particular chemical as a function of time, say in our liver. All molecules are broken down in your body at various rates. The two organs that do the most breakdown actively are the liver, which is responsible for metabolizing most molecular species in the body and turning it into bile, which eventually turns into feces, and the kidneys. And so most molecules that you take up are either gotten rid of directly into the urine or the feces, or broken down and then eliminated that way. Some molecules are exhaled through your breath. Some molecules are excreted through your sweat. So there are other exit pathways, but the liver and the kidneys are the primary organs that get rid of molecules that we've taken in. Similarly, for uptake, the stomach and intestines are the primary place you take up molecules, but you can absorb molecules through your skin, through the gut, in your eyes, through your lungs. Any surface can absorb as well. And PBPK molecules fundamentally describe the entry of molecules into the body, their circulation through the organ systems, their migration within those organs, and their breakdown and excretion from those organs. So that's the basic set of network types that we have to deal with. They all have the same structure, which is nodes that represent concentrations of chemicals, and links that represent their transport, their transformation into other species, and the regulation of that transport or transformation. In reality, almost all biological problems require multiple kinds of networks working together. They'll include some level of chemical transformation. They'll include some level of activation, inhibiting, and signaling. They often will change gene activity, and they always will require transport of materials between multiple locations. And so any real complex biological problem will have all of these classes of network at one level or another, although very often we'll simplify things by assuming that some of these processes are operating in the background, just the way that when your car engine is working, you don't exactly care about all the details of how the engine works. You just care that it's providing motor power to your car under the control of your accelerator and your brake. So we are going to be in this class using the Antimony model specification to allow us to build dynamic networks in Computational. And we can do this using Qt, or we can actually import these models from outside. Those models can be loaded into individual cells or into the simulation as a whole. Once loaded, they're converted to ordinary differential equations and then solved by an SPML solver library inside of CC3D. We can specify our networks using other languages as well. There's a language called MabBus that's directed primarily at stochastic Boolean networks used to describe gene regulatory networks. There are also direct SPML solvers, and there's a language called CellML, which is another language network specifier. And so you'll see in Twit, everything is under the name SBML, System Biology Markup Language, because that was supported by Computational before Antimony. Again, I emailed you a cheat sheet, which has all you need to know about Antimony in one page. I just gave you a link to the documentation as well. And so the first thing that we're going to do is load some code that will allow you to run Antimony models inside of Computational. And inside of the student materials demo for lecture 13, there's a folder, Antimony demos, and it's called Tellurium. And I would like everybody to take a minute and get that running on your, at least loaded into Twit, okay? Demos, lecture 13, Tellurium. For some reason, it's in a subdirectory. There we go. Okay. Please, if you need to, download the demo Tellurium from the student materials directory and get it running inside of Twit on your computer. So normally I don't tell people to use code without understanding how it works, but for the rest of the class, which is not very long, I will ask you to indulge me. If we open up the Tellurium steppables here, you'll see at the top of it, it says type Tellurium model here. And then you'll see in lines 4 through 15, it says model equals, triple quote, equations, parameters, initial conditions, quote. And then in line 18, it says variables, and the variables are named X, says plot them in red, Y, plot them in blue. And so what this allows us to do is to type an Antimony model definition, run it, and see what it does. Now, what the CompuCell code is that allows you to do that, it's not that complicated, but I don't want to go into that right at the moment. I want to concentrate on that, the antimony. Let's run it. I'm actually going to close. I'm going to close player and relaunch it. It seems to have an issue when I. All right. Well, that wasn't very pretty. It's a sizing issue here. Let me get this fixed. Wow. Okay, let's get things reorganized a little bit. Well, I don't know why the axis labels are such a mess, Juliano. That seems to be new with 4.0, 4.4. Okay, what you should see, and I hope yours looks better than this, is you'll see a plot which has two lines. You'll see a cell field which has nothing in it. And so I'm going to hide the cell field because it doesn't have any cells. And this is the program that we've written plots the concentrations for X and Y for that model that we just defined. And so why don't people try running it and make sure that it works? Does it run for everybody? Elmer says he gets an error.
I'm getting an error also.
Are you also in a Linux system?
No, I'm on Windows.
What's the error?
It says error, attribute error, SBML solver error line 801. And it's from a file called SBML solver helper.py. I can share my screen if you want to see.
I have the same error actually.
Okay, so that unfortunately is not something that we are going to be able to deal with now. That is an install problem with your version of CompuCell.
Gotcha.
And I'm not sure what to do about that. I think to get that to work, you're going to have to reinstall CompuCell. And I believe that I was working with Gabriel on this, and he had installed a different package after CompuCell. And that different package had done something to the Python libraries, which caused an incompatibility. And so you had to reinstall CompuCell. So I'm sorry about that. That's a newish problem. But I think he installed PyTorch, and somehow PyTorch clobbered that particular part of CompuCell. When he reinstalled CompuCell, it didn't clobber PyTorch, but the other direction there was an issue. So I will ask for your indulgence for the rest of class today. This is one of the reasons that while NanoHub isn't perfect, when I'm teaching a large class, I typically use the NanoHub install, because that way at least everybody has a uniform experience. So we don't wind up having to having to debug that, but I'm afraid that specific error I've seen, and it's a result of a Python conflict for the library for Tellurium. If you're running the older version of Computational, you're running at 4.2.5. Is that right? 4.3.
How would I check that?
If you go up to help, and it'll tell you what the version number is.
I'm using 4.3.2.
Okay. So when you upgrade to 4.4, that will fix that. So I apologize for that, but I don't think, and the install is relatively slow. So why don't you listen for the remainder of the class, which is not that long. And then if you and Elmer want to meet separately and go over it to catch up, we're happy to do that. Okay. So, and I'm still stuck with the plotting not working in context of Zoom. So we have this. So you should get something that looks like this, which it does, modulo the axes labels for me. And if we look in Antimony, what we'll see, and I'm going to switch back to just sharing the PowerPoint for the moment. I think you're only seeing the PowerPoint, yes? Because I need it to be a little bit bigger so that you can see more easily. Is that we are running a chemical reaction here. Species X turns into species Y at a rate B times X, that's mass action. Species Y turns into X at a rate Y times C times Y, that's mass action. And happens that the equilibrium ratio between these two, which is X labeled X star, is C over B. And so we could go into our code here. We'll see in line 6, chemical reaction, X goes to Y, rate B times X. Line 7, Y goes to X, rate C times Y, the value of B and the value of C, and the initial value of X of 10 and Y of 0. For the people who have it working, does anybody have it working? Did it run for anybody, or did it fail for everybody? Ibrahim, it worked. Why don't you try changing the value of C in your code from 0.01 to 0.1, and then run it and see what happens. If you do that, what you should see, and I can do that with you, I'm going to change C from 0.01 to 0.1. I'll hit save. And I'll run my simulation again. And it actually looks very similar. What will change is that the value of X and Y, when I'm equilibrating, will change. Before, I wound up with 10 times as much Y as X. Now, with these two parameters equal, I'm going to wind up with equal amounts of Y and X. Okay. And if you like, you could play with B and C a little bit and see how the rates of reaction and the concentrations change. I'd like to now walk you through, we don't have very much time. In the last 20 minutes, I want to walk you through the antimony language. How do we specify chemical reactions? First, it's important to know that all antimony variables, parameters, and rates are available as dictionary entries. Antimony models can be loaded either as free-floating, which runs in the simulation as a whole, or separately in cells. And we can reference any variable. If we have a variable X in antimony, then if it's a free-floating model, that variable would be referenced self.sbml.model X. If it was loaded into a specific cell, it would be cell.sbml.model X. And so communicating between the subcellular or supercellular network and the computational layer is just using a dictionary. I can read and write to dictionary entries. So now let's think about an example, and I'm going to think about a very simple example. This is from 2 years ago during COVID, and we're going to think about a population of uninfected people, T, or uninfected cells, T. We have an amount of virus in the environment, V. The probability of becoming infected is beta times the number of uninfected cells times the amount of virus, and they become infected. And I'm going to walk through the language. And again, in your cheat sheet, all you need is that middle column of page 1. Everything you need is in that one column. Again, what I like about antimony is it's very simple. Okay. This is a bit more about that connection. If you want to make that connection between antimony and Computational, you go to the Twit SBML solver menu, and it has all of the functions that you'll need. But let's just think about antimony syntax and for the last 10 or 15 minutes of the class. Antimony is a string. It's just a sequence of alphanumeric characters. Typically, it has carriage returns in it, and that's why it has to be in triple quotes. Python uses triple quotes to define strings with carriage returns. Model specification here is in yellow, and it's important to note that antimony does not write a program, it writes a model specification. That specification is processed, it's compiled, and that means it doesn't behave quite like a procedural program. It does some things that are a little bit different. The first thing that we're going to learn is probably something that doesn't do anything, is the comment character instead of a hash. Actually, hash now works in antimony. Double slash is the classic comment character in antimony. Semicolon is the delimiter. So you can either use a carriage return or a semicolon to define a line break. So I could put everything in one line. If you're used to C or C++, you'll know where semicolons come from. The fundamental concept in antimony is chemical reaction. If the chemical reaction is T goes to I1, I type T dash arrow I1. So I just draw the arrow, and then the rate, which normally would be written on top of the arrow, is written after a semicolon. T goes to I1, semicolon, beta times T times V implements this rate law. I can name a chemical reaction E1, and that will make the value of beta times T times V available. I don't have to name reactions, but it's good practice to name them. Notice that this one arrow, just as one arrow implied multiple ODEs, this one arrow generates a rate law for T and a rate law for I1. So it generates two ODEs. Just repeating that. T goes to I1. I type this as T arrow I1 semicolon beta times T times V. That generates the two ODEs. A more complex model would have many arrow diagrams. Each one turns into one line in antimony. E1 is the name of the reaction and also the value of the rate. T goes to I1 is what transition is occurring. Beta times T times V is the transition rate. That's all there is to it. If we had a complex chemical reaction, like 3 molecules of A plus 5 of B go to A plus 2C plus 9D, we type that just as it's written. 3A plus 5B goes to A plus 2C plus 9D, semicolon, rate K times A cubed times B to the 5th. And that will automatically generate a rate equation for each species, A, B, C, and D. And it will automatically take care of the fact that there are 3 disappearing here, 1 being created here, 5 disappearing here, 2 being created there, 9 being created there. And so for chemical reactions, no matter how complicated they are, antimony is extremely convenient. One thing to remember is if I'm exponentiating, antimony uses the caret instead of the double star for exponentiation. Sometimes I have a molecule created out of nothing. I have a source. Nothing goes to A at a rate K. I can write nothing goes to A at a rate K. So I can have a dangling arrow. This creates a molecule from nothing at a fixed rate. I can have a decay. A goes to nothing at a rate K times A. A goes to nothing at a rate K times A. So you do everything just the way you'd write the arrows. If I want to, although I don't recommend it, I can write first order ordinary differential equations directly in antimony with a it shows you I'm tired today. An apostrophe. T apostrophe equals beta times T times V implements the ODE dt dt equals beta times T times V. Notice in this case, I don't get 2 equations for 1. I'd have to write a separate equation for T being destroyed and 1 for I being created. This is functionally equivalent to writing nothing goes to T at a rate beta times T times V. Generates the same thing. There's a good reason to use the arrow notation. If I use the arrow notation, I can always translate it into ODEs. However, if I give you a set of ODEs, you cannot back out what the arrows were. You lose information when you go from the arrows to the ODEs. And so you have more information, a more understandable code when you use the arrows. Okay. If I have multiple reactions involving the same chemical species, they add up. So nothing goes to T, T plus V goes to I1, I1 goes to D. Everything adds in the ODEs. I have to specify the values of all parameters in my model. If beta and V are parameters, and I don't specify the values, antimony will generate an error. It's always a good idea to include the units as a comment and say, this has units of TCID per day, this has units of molecules, this has units of something. I can use scientific notation 1E7 for a value if I want. For initial values of variables, I can assume a default of 0. but that's a dangerous thing to do. So you should always specify the initial values of your variables. But Antimony will not generate an error if you fail to do that. And that's it. That's all you have to do in Antimony. And so the exercise, perhaps we can do, we only have 3 people left who've hung around. And I guess Elmer, you don't have code that works. So Mike and Ibrahim, is your code working, or do you also have it not working?
It's working for me.
Yeah, my code worked also.
Okay. So what I would suggest you do here is take this tellurium model that we have here, which has 2 rate laws, and change it to be a single rate law. T goes to I1. And the only thing you have to do is in the line where it says variables, here we had X and Y. Here I would type T and I1. And you can try that. See if that works. Now, there are a couple of bells and whistles. I'll talk for a minute here. There are a couple of bells and whistles for Antimony. It allows you to do things like compartments. It allows you to have events. It allows you to do a variety of other things. But in general, for the things we need in Computational, you don't need anything more sophisticated than what we've seen here. The key thing is to know if I see a particular arrow diagram in a biological context, what rate law do I have to write down? And that's not a question of Antimony versus some other modeling language. That's a question of how do I interpret the biological notation in terms of mathematics.
Error, do you think you can help me find it?
Okay, let's see. Let's see what you got. I'll stop sharing. We can
Yeah, I believe I copied it. Oh, this should be capital B probably. That's probably the problem.
yes, it does. So it does care about capitalization. Okay, let's try running that.
Can you still see my screen?
I only see Twitter. You have to reshare if you want to show play.
Now, can you see?
Yep, there we go. Okay, so it's running. Not the most exciting model in the world. All the cells get infected and that's done. Okay, anybody else? I guess the only person who could, the only else on the call at the moment would be Mike. Did this work for you or not work for you?
Whenever I try to load it in the player, it just crashes the player. I've noticed that has happened with CC3D before when I try using it with Zoom, so it might just be that, but I'm not sure.
You want to try a screen share and see what happens? So you've got it typed you've got it typed in Twitter.
I can share what I have in Twitter.
Okay,
Maybe you
let's
could,
take
like,
a look.
see if there's something that stands out.
So one thing that you may run into with Twitter Antimony, which is like the XML, is that if there's a syntax error in the Antimony, player may crash.
Okay.
Computational is very good at catching errors in Python. It's less good at catching errors in.
You've got a semicolon after the V. Yeah.
Right there. Yeah, there. Thanks. That might be it.
I think it's also, you've got a lowercase v probably. I can't tell. Is in line 13, is that upper or lowercase v?
That's uppercase, yeah.
Okay, fine. It's small for me. Okay, and is it still crashing on you?
Yeah.
And it doesn't throw an error there.
No, yeah, it just crashes.
So that might be a Zoom interaction.
I think you need to add like red and blue for your variables.
Let's
Oh.
see. Let's go back to it.
Right.
Let's take a look again.
Oh yeah, I think I missed that.
Let me.
Okay, yeah.
Oh yes.
Yes.
Under variables, you have to have T : red, yes, and then comma. So it's T1, no, T : and then the color, comma, I1 : the color. There we go. Try that.
Okay. There we go. That works. Can you see
Okay.
the?
No, you have to reshare it. For that.
Okay. There we go.
Okay, good.
Yep.
So you've got the basics working for two people, and next time maybe Juliano can give a couple of very simple exercises that would have been the other ones that we were going to do in class here. The rest of the class that I had planned, and it's nothing very complicated here, was to do a few exercises where we built some slightly more complicated chemical reactions. And so it's really just to explore the way chemical reactions work. But fundamentally, if you've got the syntax for one arrow working, and you know how to change that one line where you have X comma color, you can write an arbitrarily complex network. And so that's where we're going to stop for today.